Now, after the DNA tumor viruses, the DNA oncogenic viruses, now the RNA oncogenic viruses. There are the three kinds of the RNA oncogenic viruses. There were about 5 or a little more species of the DNA oncogenic viruses, but here we will see that what are the RNA oncogenic viruses, what are the RNA viruses which are capable of cause the disease and cause the cancer after the infection. These are the oncovirine viruses, Lenti Viruses, very important from the human point of view, it causes the immunodeficiency syndromes, and then the Spuma Viruses. We can see them one by one, Spuma viruses, that how they are causing the disease. First of all, the mode of action of the RNA viruses, how the RNA viruses are capable of cause the tumors. As you know that RNA viruses, the reverse transcription is very important from the RNA viruses point of view. Even we in the molecular biology, we taken the idea from these RNA viruses for the reverse transcription, and these days we are using this in the genetic engineering on the large scale. So viral RNA genome, first of all, is the reverse transcription, and the viral RNA genome will be converted through the virus transcription by the DNA genome. Of course, it has to be converted into DNA before, and then the integration. Then the viral genomic RNA will be converted. After that, the DNA dependent RNA polymerase, which is use the RNA, the host machinery, RNA pol 2, and it will be converted into viral genomic RNA. Then for its translation, transcription, messenger RNA will be converted through the splicing, and then messenger RNA, the viral protein, and then the virus will be formed, and this is using by the viral host machinery. This is the life cycle of the RNA tumor viruses. We will take the example of one by one the different viruses, the RNA viruses we have just studied, the oncovirine, Lenti Viruses, and the Spuma viruses. We will see them one by one, that how they are capable of cause the disease and what kind of the cancers are caused in the animals or in the human beings, which is caused by the specific type of the viruses. So oncovirine was first of all discovered by the Ross sarcoma virus in the slow neoplasm, and it was discovered in the chickens. In the chicken, it was causing the Ross sarcoma, the cancer, and first of all it was discovered, and it was caused by the oncovirine. Human T cells lymphotropic virus is also, this is called the human T cells lymphotropic virus. It is the oncovirine. Here is the oncoviruses, RNA tumor oncoviruses. It is the function, and it is its structure, the morphology, how they are capable of replicating inside the body. RNA tumor virus, first of all, we have seen it, the same phenomena as structure, the same morphology as we have already studied the life cycle in the DNA viruses. Fusion and entry, the virus is capable of the, first of all, its coat, and here is the DNA or RNA. Of course, in this here it will be the RNA, and it is the capsid RNA, and then the coat, it will be had to be entered inside the body. The coat will remains outside. Here is the RNA, which is will be capable of the, there is the converted after the replication, it is the integrated inside, and then viral genomic RNA, translation, transcription, and then after the translation and transcription they will become together, and the assembly. After the assembly of the virus will be come out and mature outside the body, and then the virus is again capable of, in this way it is cause the disease inside the cell. So after the disease, how the virus, how it is converted into the cancerous cells. Then are the Lenti viruses, Lenti Viruses, important from the human point of view, that they are causing the disease of HIV. We have already studied in detail the human immunodeficiency syndromes, because in this kind of the viruses, the human immune system becomes confused, and it is incapable of recognizing its own T cell, its own immune system bodies, so it is going to kill its own body. The human immune system produces antibodies against its own immune system. Therefore, it is very difficult for treatment of such kind of the disease. So Lenti viruses have long patent period. They are capable of reside inside the body or reside outside the body, latency. They are hidden inside the body. Then disease of the ungulates is also there, and then HIV, that causes the AIDS, immunodeficiency syndrome, acquired immunodeficiency syndrome inside of the human beings is the AIDS. So RNA oncogenic viruses, the structure of the RNA oncogenic viruses, Surface envelope protein first of all is there, and then the transmembrane envelope proteins, membrane, matrix proteins, capsid. We have just see the life cycle. The capsid will go inside the body with the RNA, and then RNA genome, and then the integrase is also present. Then integrase, that's why it is integrated, and then the reverse transcriptase and the protease. Now the Spuma viruses, Spuma virini, Spuma viruses, which are also capable of cause the disease. Because of the infection caused by the Spuma viruses, the appearance of the cells becomes foamy, and the syncytia formation of the G and multinucleate cells are also there, from which we can diagnose that this is the disease, or this is the infection caused by the Spuma viruses. Human foamy virus, it is also called as the human foamy virus, and the simian foamy virus, because of course they are causing the disease in these animals and also in the human beings, so it is also called as in these names. Here you can see the morphology of the simian, human, the Spuma viruses, and the disease caused by it. It is very prevalent from the picture given below.